Hello and welcome to this video on PCR types and what they do. PCR, or polymerase chain reaction, is a genetic test that measures how much of a given single gene is being expressed in a tissue, cell, or similar small sample. It can be extremely accurate, efficient, and powerful, or a blunt tool that keeps working despite hiccups. PCR is efficient and cost-effective overall compared to some of the older molecular tools available. It's able to copy and amplify small signals from DNA or RNA. It combines some of the most fundamental parts of genetic research, that being the complementary nature of DNA, and that you can then replicate DNA based on this complement. This can be done repeatedly throughout a number of cycles in the PCR system. This means you can get an exponential growth in the signal strength. In order to get this to happen, you need something called a primer. A primer is a short section of DNA that matches with your DNA or RNA. This is what allows it to be amplified. By matching the two, you get it to become confused and think the DNA needs to be replicated again and again. By choosing primers that specifically match to a DNA sequence, you can be very selective, but they need to be both a forward primer for one side of the DNA strand and a reverse primer for the other. You then need some sort of probe. The probe is what lets you know that the DNA is being replicated. If you were just trying to measure the amount of DNA in solution, you could use something like a nanodrop. That would use an amount of light shone into the sample, and the amount reflected back would give you an idea, but that's also prone to becoming contaminated. The easier way to do it, and especially for some of the methods we'll talk about for PCR, is this probe. The probe signal becomes stronger the more the DNA is replicated. Therefore, adding the appropriate probe allows you to give a better, stronger, and more consistent result. Different options available include radioactive compounds, colored compounds, fluorescent compounds, and chemiluminescent compounds. The choice of which one you use depends on the system and the nature of what you're trying to investigate. Once you've chosen this and you've begun replicating the DNA, the probe does two things. One, it allows for visualization. That is, if we're looking at a fluorescent or colorimetric product, you're going to get an intensity increase. Secondary to that is that because you have a specific primer being used, and that primer ensures that that label or probe goes into only the copied DNA sequences, you don't get a significant increase in off-target DNA or gene sequences. This means you can be very specific, even if the primer itself isn't as specific as you might want. You should still get a consistent and reliable result. With this basic background information given so far, you have most of the common information to PCR as a whole, but there are many different kinds with strengths and weaknesses. The ones we'll cover in this video are real-time PCR, which is quantitative PCR or qPCR, Reverse-Transcriptase PCR, Multiplex PCR, nested PCR, high-fidelity PCR, fast PCR, hot start PCR, GCRich PCR, long-range PCR, and arbitrary primed PCR. Quantitative or qPCR is the most common and regular form of PCR used. Fundamentally, it quantifies the amount of DNA at the start of the sequence, and then measures it right throughout the process. This gives you something of a curve, and that curve will tell you how many copies are being created over time. It has a relatively high degree of precision because you can see that change over time, and often uses a number of different fluorescent dyes, cyber green being a very common one. Quantitative PCR is also sometimes known as real-time PCR, and that in turn is abbreviated to RT-PCR, but RT-PCR is a misnomer, as RT is used for the next version of PCR, otherwise known as reverse transcription PCR. This works in a similar way. It amplifies the DNA or RNA. The difference between this and qPCR is that it begins by amplifying the cDNA from the DNA or RNA using a reverse transcriptase in the PCR process. This is particularly useful if you're looking at trying to find particular genes in DNA or RNA. Consider what you would be doing if you're looking at forensic evidence as an example. Trying to eliminate certain parts of the population based on a limited sample from a crime. You could use this method to locate certain exons and introns in genes that would then allow you to look at the wider population and see who would or would not likely have these particular mutations. Next, we'll look at what happens if, rather than looking at a single gene, you're trying to understand multiple genes simultaneously. This is known as a multiplex polymerase chain reaction. The multiplex part of this describes how you attempt to amplify multiple genes simultaneously. The difficulty with this particular approach is that you need to use multiple primers at the same time, but most primers require different temperatures to achieve that result. This means that you need to find a way around the bottleneck, that is, the temperature-mediated changes. All the primers have to be designed so that they all work as close to the same temperature as possible during the PCR cycle. This is the weakness for multiplex PCR. The strength is that if you have a limited sample, such as archaeological or anthropological remains, it is possible to get a lot more data out of a much smaller sample. If, rather than looking at getting multiple possible genes or data out of a single sample, but rather you're focusing on a single particular gene, but one that is large, you should look at nested PCR. Nested PCR uses several repeated processes of going through the polymerase chain reaction. But what you're looking to do is to extend one gene that's quite large, but it requires two pairs of primers. This will generate a large DNA product in the end. The catch with this is that you have to know what you are looking for. That is, you need to be familiar with the target sequences of where the primers have to go to be active. Unlike the others where you can have some degree of leeway in designing the primer, or you might be looking for what's in the genome. This needs to be well understood and established before you go in. If you need to be even more exacting than what occurs in nested PCR, you should be looking at high fidelity PCR. This is essentially a process that is a lot more exacting and requiring of knowledge than the others have been. It's often used for cloning and similar applications. You don't want to make mistakes in transferring or copying DNA sequences if you're trying to make a clone. The entire point of the clone is that they should be exactly the same. It offers the best possible performance with short procedures, and generally speaking, smaller gene sequences. Because of this, the high accuracy and the fact you're working with small gene sequences, you tend to get better yields. This means that unlike other processes, you don't have to go for as long in protocol time, meaning you can get more results quicker, and often on par or better than longer sequences that aren't as accurate. Fast PCR is a bit different but more or less the same to classical PCR or the qPCR we mentioned. The difference is how long you run the reactions for, and in short, in order to run a fast PCR, you do exactly what you would with conventional or qPCR, but you have to have the most optimized, effective, and reliable enzymes and PCR mixtures. This means you need to be far more exacting and demanding, and while you won't get any better results than in qPCR, you can get them quicker, at the expense of a lot more time, effort, and preparation, and sometimes results can be unreliable if any mistakes have been made in preparation stages. Hot PCR is, again, similar to qPCR. The difference between it and the hot PCR procedure is that you are following the same general principles, but each of the stages that are used to provide that curve and growth in the signal going back to the PCR machine is extended and made more exacting. This allows you to take what would otherwise be broad bands of activity and break them down into smaller, more modifiable and controllable situations. Think of it as the way in which a car accelerates. An automatic car can have you simply put your foot down on the accelerator and it will take off, gaining speed the entire time. A manual car would require that you change gears at regular intervals, but you have control over how high the revs get, at what speed you change gear, and so on. This is the difference between the hot start PCR and qPCR. Hot start allows you to choose exactly when certain shifts, such as temperature, occur. This means you have absolute control over when certain things occur, such as inhibition of polymerases. This means that you can make sure that the proliferation of the DNA strand and how many times it's copied, when it's copied, and what occurs is under absolute control by the machine. This greater degree of fine control is what lets it specifically bind the primers to the right DNA sequences at the right temperatures, and then to denature them. This gives a very, very specific result. This can then be further refined again by addition of certain agents that would block DNA binding at lower temperatures than the primary intended range. This means that you can ensure that only the desired ranges as far as temperature goes work. GCRich PCR is another version of PCR yet again. Like the others, it's designed to be more refined and specific, and in this cases, it uses a variety of polymerases, and these polymerases allow for unique, particular delivery of certain templates. Now, some templates, particularly if we're talking about those that are based on a primer sequence that's ubiquitous, have a lot of difficulty in targeting where they should be and how they should be. If not the more difficult and ubiquitous genes, then it can also work for those that are much larger, up to 5,000 kilobase pairs. So you now have the ability to target long and difficult to access genes that could otherwise be confused by using the particular polymerases that are in this. The downside to it is that it is a proprietary product, unlike others which are at least somewhat proprietary, but they work within a larger framework, a framework that is generally developed in public research. Long-range PCR takes the idea of GCRich PCR and makes it even better. This is used for DNA that is more than 5,000 kilobase pairs in length, and it uses yet another specialty and proprietary DNA polymerase. It is a special version of the high-fidelity PCR. Finally, we wish to talk about something known as Arbitrary Primary Chain reaction. This is a sort of fingerprinting technology. That is, it allows you to use a small short primer, which generates varying lengths of products from the whole genome sequence. Once this is done in PCR, and you get your initial results, the products are then separated via gel electrophoresis. This technology can be useful if you're trying to figure out whether or not, for example, a bacterium is in a related family or the same family as another sample you've taken. It allows you to randomly select a series of DNA sequences from in there using that primer, separate it in the gel and compare the two gels, which should read somewhat similar to a barcode. The reason the arbitrarily primed PCR is reliable and replicable is that it should bind to the best match in the genome. This means that you should be able to take the same sample, run the same reaction again, and be able to see the same results. The results should be contingent on the intensity of the gene expression, which means you get not only a quantitative but a qualitative assessment. The weaknesses to this particular approach are that you need to operate under the same circumstances for each set of runs. This means that if you vary either the setup for the PCR or the primer sequences used, you can't compare results between samples. This has been a very broad summary of the various PCR methods available. Obviously, each one requires much more detail to fully understand, but seeing what a lot of the options currently available are should give you some idea as to why and what is chosen at different times. And again, there's a lot more detail that can be had, but not that can be fit into a video of this size. For now, thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.